respected delegates, Dr. Anil Thachil, currently working as Head Bacteriology and Mycology Section, Rollins Animal Disease Diagnostic Laboratory, Assistant Professor Population Health and Pathobiology. After his graduation, he did his post-graduation studies in clinical medicine from College of Veterinary and Animal Sciences, Kerala Agricultural University, India. He did his postgraduate study in veterinary medicine, infectious diseases from College of Veterinary Medicine, University of Minnesota. His research interests are infectious diseases. He interested in proteomic and genomic characterization and fingerprinting of bacterial pathogen, including Salmonella, E. coli, Streptococcus species, Clostridia, Ornithobacterium, etc. Another major area of interest is the changing epidemiology of antimicrobial resistance and dispersion of successful clone of multi-drug resistant bacteria. With these few words, I'm inviting Dr. Anil Thachil to the webinar. Thank you, sir.
Thank you, Justin. Now I invite Dr. George T. Oommen, former professor and head of Department of Livestock Products Technology and Meat Technology Unit, Kerala Veterinary and Animal Science University, and Dr. Deepa Jolly, Assistant Professor, Department of Veterinary Public Health and College of Veterinary and Animal Science, Mannuthy. Now I am handing over this session to our chair and co-chair, and welcome Dr. George T. Oommen for opening remarks.
Hello, good evening to you all. Can you hear me? Hello, good evening to you all. We are running short of time. So, welcome Dr. Anil Thachil, our alumni. And you are going to speak on the myths about meat diet and zoonosis in pets. And there are two different topics of interest. And you are touching, because a lot of people ask about, they have a lot of questions about the meat diet, telling if meat you will die because of eating that. So that I think he'll be covering all those portions, and as well as the zoonosis in pets. Dr. Kumar now touched a new, it's not a new bacteria, but C. difficile. It may be present here also because it goes unnoticed because of lack of surveillance and monitoring and all. Maybe there, but we have to do some work on that one and all. So I welcome Dr. Anil Thachil to carry on with his talk and elaborating more on these topics. And let me congratulate the IVA Mannuthy unit, Mannuthy Veterinary Ecology unit, the Meat Technology unit, and Association of Meat Scientists and Technologists. And thank you for inviting me to this one. Thank you. Dr. Anil Thachil.
Okay, so let's discuss about the myths or misconceptions, about meat diets, especially in US, and how it is linked with the increased zoonosis in pets. So before starting my talk, two disclaimers. One is I am a CLSI member, and I'm also a North Carolina state employee, so dealing with some of those regulatory overseeing with FDA and USDA. So I graduated from Mannuthy, and then I have had a short tenure at the Animal Husbandry Department, and then I moved to University of Minnesota for my Ph.D. And University of Minnesota or Minnesota State is #1 turkey producer in US. So they produce around 2 billion pounds of turkey every year. So then I moved to Iowa State University Diagnostic Lab for a short career. And Iowa State is like the #1 pork producing state in US. And the overall pork production is going up in US, and in 2019, we had seen a 10% increase in the export, and US export now around 7 billion pounds of pork to the other countries. After my tenure at Iowa, I moved to Animal Health Diagnostic Center at Cornell, and it's the biggest veterinary diagnostic system in US. And at HDC, we test around 10 lakh, individual diagnostic testing every year. And then it's also, it's located in New York State, and the New York State, people maybe, heard about New York City, but you won't believe, like, the New York State is like the third largest producer of milk in US, after California and Wisconsin. So it's also an agricultural state, actually. And after that, I moved recently to North Carolina diagnostic system, and where we North Carolina Agricultural Sciences Center, where we test currently around 7 lakh individual veterinary diagnostic testing every year. So it's a very big diagnostic system, and we are also having the capability to look into the human food and drug, also. And the outline of my talk today, I'll be discussing some of those misconceptions about meat diet, especially in US, and especially at the human-animal interface dynamics. So I'm not like touching any sensitive things, but we, unfortunately see a lot of misconceptions about raw meat diets. And because of that, we see a lot of, increased zoonotic infections. So I'll also discuss about the role of meat diet in causing, pet foodborne infections. And I also briefly touched the recent approaches to contain some of these foodborne zoonotic infections, and also like discuss some of those role veterinarian as well as veterinary diagnostic labs can play in public health to control the zoonotic diseases. So I like that, the topic of the webinar, the human-animal interface and the recent approaches. As we all know, like, dogs and cats, they're the, human's best friend. So we see like, increased number of, families or households are having pets now than ever before. And we see more than 67% US households, own at least one pet. And during these stressful times of COVID, and even other stressful times, we see more, pet adoptions, in US, and we all know that, animals can relieve in our stress. So there is an increased demand, to, own pets now than ever before. And humans now, have more closer interaction with these pets than ever before. It's all over the country. We see like more and more indoor pets now, and because of that, we have an increased interaction with the pets, on a daily basis. So we also improved a lot in our animal welfare activities. We moved from caged chickens to cage-free now, and even free-range organic and pasture-raised chicken. So we made a lot of progress in the animal welfare. And we also see like, increased urge to provide, natural environment, so for the pets. So people are like, back to nature, and we want like, everything like, more natural, and try to create, as much as a natural environment for all animals. Maybe because of that, we also see, an increased drive by people to, feed raw meat diets, for these dogs and cats. So my first myth or fact, we used to see a lot of people telling that feeding raw meat makes my dog or cat happy. Okay, so this is something like to be looked into, and we need to see whether there are any scientific support to do that, or, are there any risk factors? Okay, people oftentimes, ignore the risk factors behind it. Okay. So we all know that, social media plays a big role now, and we also see a lot of drives, in the social media for back to nature. People are after natural foods, try to see like, how we can, get more close to the nature and protect nature. And because of that, we also see like, a lot of raw meat diets feeding, and it's really alarming. And it causes a tremendous increase in zoonotic infections in dogs and cats in US. And also we see a lot of public health risk. A lot of people are also getting infected either from pets or these pet diets or raw meat. And there are no scientific evidence to support any of these health benefits because of feeding, raw meat diets to pets. But people are somehow convinced, about this raw meat, diet feeding to dogs and cats. You see that in the market, there are lots of raw dog diets now. It's increasing day by day. You can see like a solid, dry food with meat ingredients to raw meat to semi-solid meat, even chicken jerkies. There are like freeze-dried, products. There are like pig, ear strips, can give us a treat to the dogs. So we see like increased huge variety of stuffs coming to the market day by day, to make our pets happy. So the kinds of food in the market, so we see like, the most common type or the most economical type is the kibble or the dry food. These are like pellets. And people think like, that is, kind of like, any bacteria free. But we see like, even those can, get like, zoonotic bacterial contaminations. And we see like, canned wet food, again, a higher risk for zoonotic infections. Semi-moist food like pork chops, burgers, other meaty foods, and raw pet diets, usually include a combination of uncooked meat, bones from animals, fish, or poultry. Oftentimes, many of these ingredients are, not considered fit for humans. That's why they are including it in the pet diets. And sometimes, we may end up sometimes seeing, rejected carcasses or even medicated animals, coming to the, pet food diets. We got a, case a few years back about a phenobarbital poisoning in some pet foods because they included some animals, after euthanizing them, with the phenobarbital. So, but anyway, the quality, oftentimes, may get compromised. So the sale of pet foods are soaring. So we see a huge demand, it's always growing demand for the raw pet diets. And a staggering number of people are feeding their pets, either with the commercial pet diets or butcher offcuts, and they claim it as fresh food for their dogs. And we see like more diverse range of products, including, freeze-dried raw foods. Almost all, foodborne infections we at least see in the diagnostic lab, are linked somehow with people feeding those raw meat diets. And we also see like, an increased pet food recalls in the last, couple of years by FDA. That also indicates that, we have more risky foods on the market, and the new, the raw diet, trend is more risky. So if you look at the World Health Organization list of zoonotic diseases, and so these are the zoonotic list, diseases they listed out. And out of, 6 out of 10 infectious diseases in people in general are considered to be zoonotic. And in that list, although I'll be more, interested to talk about Salmonella, that's listed, as a World Health Organization zoonotic disease. And I also have one slide each on brucellosis and Campylobacter. And these are the 8 zoonotic diseases of most concern, in the US. In 2020, they updated the list. And you see like, the salmonellosis and brucellosis, are listed among the top 8 infectious zoonotic diseases of concern in people. And again, we see like, salmonellosis and brucellosis, in our dogs and cats. And foodborne bacterial infections, so what are the most common foodborne bacterial, infections we see, in pets in US? I also, and I already mentioned about Salmonella. So we did a study, or FDA did a study. This was like, 6-7 years ago. And they looked at like, a lot of commercial pet diets in the market. And they find like, out of 200 samples, around 7% of them were like positive for Salmonella, and around 16% of them were like positive for, Listeria. This is like, a few years ago, and this, study actually realized that, we need to look into those, pet food diets more closely and increase, the standard of those, available to the public. So the most common pet food infections, I told that, we get it from the raw pet diet, and these pet diets are equally dangerous to the, client owners too, or the owners of the pets. People can get infection, and also the dogs can get, infection. And the most common, raw diets are meat, poultry, milk, eggs, that may not be cooked or, treated, to remove any of this harmful bacteria. And the most common, foodborne bacterial infections we see are Salmonella, Listeriosis, then Campylobacteriosis, and we also see occasionally, Clostridial infections. And there are a couple more, which is not highly prevalent, like Yersiniosis, very rare, but these are the 4 most common foodborne, diseases we see in pets. And if you look at the human, diseases, like the human foodborne diseases, in the CDC chart, the 4, highlighted here, Salmonella, Clostridia, Campi, and Listeria, they are in the top priority list for humans as well. Okay, so these 4 diseases are not only, we see in pets, so it's more risky, in humans. And if pets get infection, humans will get infection, as well as vice versa. So talking about salmonellosis, as Salmonella is the most common foodborne bacteria in humans. And the name of Salmonella is named after Dr. Salmon, is a Cornell alumni. And we see like more than 1.5 million illnesses reported in US or diagnosed as Salmonella. And one peculiarity is like most of these infections are self-limiting, or it will go away in healthy individuals. And but we still see like some of those Salmonella are more pathogenic, and we see like increased hospitalizations. Every year we used to see at least 35,000 hospitalizations. So these are hospitalization is very expensive process. And we see like, mortality around 450 every year. And we know that there are more than 2,500 serotypes of Salmonella. And not all of them are the same, but there is a high zoonotic potential for all Salmonella serotypes. And also another concern we have is the antibiotic resistance. Okay, and Salmonella is getting more and more resistant nowadays. And we see a lot of multi-drug resistant Salmonella causing, more, cases than ever before. So we know that Salmonella infections are zoonotic. It can cause diarrhea in people, and most of the time it is mild or self-limiting, but it can turn to severe or sometimes, take life. Dogs and cats, mostly they are asymptomatic, but also they can get sick. And we see sometimes, decreased activity, vomiting, GI issues, sometimes, fever, and in very acute cases, we used to see septicemia and abortion in dogs as well as in cats. And pets that eat a contaminated, food can have Salmonella transiently in their GI tract, and they may appear asymptomatic, and they can, carry the bacteria for a short period of time. So we call it like a transient stage. And the antibiotic treatment is advised only for systemic illnesses in dogs, and cats, with salmonellosis. So the reason why I will, discuss that in the, next few slides. But it's not advisable or to treat dogs and cats which are asymptomatic, or you may find some salmonella in their feces. And the other question is like, these are like kind of like, myths. People like, oftentimes ignore this fact. The question is whether can we reliably use antibiotics to get rid of bacteria, in the gut, either in humans or in, animals. So, what is the scientific reason behind that? We cannot reliably use an antibiotic. Of course, we can use, but just for the symptomatic carriage, the antibiotics are not going to be effective, and even in clinical diseases, it may have some bad impact by use of antibiotics. So I'll tell you why in the next few slides. So that's where the CLSI comes into play. Okay, so CLSI is a Clinical Lab Standards Institute. It's a non-profit organization. We try to improve the lab testing quality, and CLSI has been the long provider of the antibiotic susceptibility testing breakpoints for humans as well as animals all over the world. And they also give guidelines for the AST methods and also quality control standards for putting up in the lab. So I'm got the opportunity to become the document development committee member. So we draft some of these documents. We recently put out a Veto 8 updated and Veto 9 document. So these are veterinary specific documents. Okay. So these are documents which provide guidelines for AST testing, and now we have breakpoints for almost every antibiotic we use in dogs and cats. Okay, so we don't have to depend on human breakpoints, like we used to before. Now we move to the veterinary specific breakpoints. And the good thing is now, there's a breakpoint documents are available, freely online. So I encourage, everybody to have this access to these documents, when you have a AST testing to treat, animals, especially in, multi-drug resistant organisms, if you encounter. So this is the CLSI website, there you can see some, tabs for the standards. Once you click that, you'll get access to the documents. Okay, so the M100 is the human breakpoint, AST document. They have antibiotic breakpoints. Breakpoints are just interpretative criteria. When you do an AST testing, you get an MIC value. Okay, but to make sure, like what that MIC value means, or to interpret the MIC value as susceptible or resistant, you need some breakpoint. Okay, and that breakpoints can get updated, and new antibiotics can come, new breakpoints can come, and this is the place to go to see what is the current breakpoint for a particular antibiotic, from a particular host or from a particular site, and the Veto 8 document, that is the veterinary document you need to go. Okay, so both are available free online, and the M100 is like getting updated every year. So the breakpoint updates can come every year, and the Veto 8, like, we frequently update it, but every 2 years or up to 4 years. Okay, so now we are currently doing like every 2 years. So this is how that document looks like. If you open a vet, 08 document, they are arranged based on organism. So, if you have a Pseudomonas infection, you can go to the Pseudomonas, section, and they arrange by, the class of drugs, the fluoroquinolones, you can choose, and then you have to look for the host you have, whether it's a dog, cat, or a horse or whatever. And then, under that, you have different, tissues. Okay, so the breakpoint can differ based on the bacteria you're getting from what tissue. Okay, if it's a respiratory or like a skin or soft tissue or a urinary, they have different breakpoints. And then, you choose the right antibiotic you wanted to use under the class. If it's enrofloxacin, and you can see to the right, the interpretative criteria, the SIR values for the Kirby-Boer technique, as well as the broad dilution technique. That's what the MICs are for. Okay, so this is a very useful, a table to interpret your, AST data, especially when you get a data from a human lab. You may get MIC value, but using this table, you can make, or customize it for veterinary use. Okay, so don't look at the human breakpoints. And most of the time, the human breakpoints are way lower. So you may think that, the organism is going to be susceptible, but it may be resistant. Okay. So that's why we need to use the veterinary breakpoints, and most of the human labs, the data you get is like based on human breakpoints. Okay. So they may say it's susceptible, but it may not be. So that's why this, data is really important. And also we have, intrinsic resistance tables in these documents, we on both in Veto 8 and Veto 9 document. There is an extensive chart of intrinsic resistance table. I highlighted only for Pseudomonas here. As you can see, these are intrinsically resistant to any beta-lactams or chloramphenicol or tetracyclines or, sulfur TMPs. That means, none of these drugs are going to work for a Pseudomonas infection. You don't need even an AST. If there is an intrinsic resistance mechanism there, these drugs are going to be resistant irrespective of your MIC, being susceptible or not. Okay. So good to be, have this table handy for treating, some of these really, bad bugs. So one thing to keep in mind about the CLSI breakpoints is like, these breakpoints are host specific, and we have only a limited number of hosts listed there. We have dogs, cats, horses, cattle, pigs, and fish. There are very few breakpoints available for fish, but still we included that. And we have like small ruminants, poultry, and wildlife. We do not have any breakpoints. Okay, so if you have a goat or a sheep, to treat for, you have to use the closest thing. The either you can go with the cattle and a breakpoint, or like any other species breakpoint. Even sometimes horse breakpoints you can use for cattle. But again, it's a food animal thing, so you need to make sure like those antibiotics can be used in the food animals. And then for poultry, we don't have any breakpoints, so you can use either human breakpoint or any other breakpoints available. And again, then our sites. Okay, we do not have breakpoint for every body site. Okay, so we have at least 3 or 4 categories. The most common one is the skin and soft tissues. That means any abscesses you get, or like any surgical wounds or any infections. You can use the skin and soft tissue breakpoint. Then for respiratory, we have a separate breakpoint. Okay, so that's very important. Okay, because some of those antibiotics can reach at high concentrations in respiratory linings. So that's why the respiratory breakpoints are different from the skin and soft tissue breakpoint. So, and urine, so urines, some drugs can reach high concentration in urine, so that urine breakpoints are higher. So, so you need to use that. And, coming to the point I want to emphasize, there are no enteric breakpoints. Okay. So that means, you cannot reliably treat, with an antibiotic to get rid of the bacteria from the gut of an animal. So all these breakpoints we have is based on the systemic, plasma concentrations. So you can achieve, the drug at that particular, standard dosage to get rid of the bacteria effectively from, the system, okay, or the blood or like any other systemic tissues, but not from the gut. Okay, so we do not have any enteric breakpoints either in humans or in animals. Okay, so we can arbitrarily treat with the most susceptible drug, but we cannot 100% guarantee that all the bacteria is going to die, because you cannot achieve the concentration at that level like in plasma to kill the bacteria in the gut. And topical breakpoints, we do have some for humans like miprozin, but we don't have anything like for animals, but you can use human breakpoints for those few drugs. And one thing in veterinary is like we have some intramammary breakpoints. Okay, so that's a separate thing. So the other factor myth this especially in US, are the meats available in the commercial stores are salmonella free? We used to see the products, at the US markets as USDA inspected, so people sometimes, misinterpret that as, this once it's USDA inspected means, you have a food inspection. So that means, it's totally void of any bacteria, which is not true. Okay, what that inspection procedure means, they, make sure like, it's a, kind of like a safe, the levels of bacterium, present may be safe for humans, but it does not mean that, it's salmonella free, or, it can pose risks to the pets if you give it as a raw diet. Okay, so people expect still, that, the commercial meats available are salmonella free, but, the fact is that, it may not be. Okay, so there is a risk in getting the commercial chicken meat, for Salmonella, Campylobacter, you may still see that, and in US, the USDA, FSIS section, do meat inspections, and they continuously sample, from the processing plants, and they test for Salmonella, Campylobacter, make sure like, the samples they collected from a 52-week moving window, comes under, their, 3 category system standards. Okay, so they call it FSIS performance standards. And usually these standards are kept so that like, people won't get infection. So the CFUs, find of bacteria find in the samples are below the infection threshold, but it it's sometimes can cause infection, or animals can amplify this bacteria and can humans. Then we in spite of this FSA standards, we still like a lot of commercial products coming to the market can cause human infections, and we see like huge recalls of this products on a daily basis. So you say like 12 million pounds of beef like infected with salmonella, and they're got recalled from the shelf. So these news are we see like occasionally, and this because of the potential for causing salmonellosis in humans or in even pets. So the question is, can we get to the zero salmonella in poultry? Okay, people wanted like zero salmonella. Is it possible? It's possible. We have SPF chickens available for research purposes, which is salmonella free, but it will be too expensive to produce it commercially. Okay, and it's also very labor intensive for controlling salmonella in a commercial system. And the FDA regulates all pet diets in US, and FDA has a zero tolerance policy for salmonella in pet diets. So that means it's very good. Unlike in humans, pet diets are supposed to not contain any salmonella. Okay, it should be totally salmonella free, but not the commercial meat in the market. So that means pet foods are relatively or theoretically should be more safe. Okay, that does not mean that you can eat the pet food. Don't do that. But still, the FDA has a very stringent policy for the pet diets. Okay. So the other thing people always wonder is like how come this dry dog foods get salmonella? Do they? So we used to see that. We used to see that even dry pet foods or pellets, with the meat ingredients in that sometimes maybe due to the manufacturing defects, they may end up with having salmonella in the dry diets even. So this is a report like came a few years ago in 2012. So human salmonella infantis infections linked to dog dry food. Okay. This became a huge multi-state outbreak linked with the dog dry food, and a lot of pet owners got infected. We have around 49 individuals got infected, and around in more than 20 states, 10 of them hospitalized. Luckily no mortality. So this is a good example of a dry dog food harboring salmonella, causing a lot of human infections. And again, in 2018, we did see another outbreak of a multi-drug resistant salmonella infection linked to the raw chicken pet food diets. Again, this time same bacteria, same Salmonella infandis again. This time it infected more than 129 individuals. But this time it was a multi-drug resistant bacteria. This was resistant to ciprofloxacin and ceftriaxone. Now these are the first line drugs used in humans. So that's why this infection is more important than any other salmonella infection. A lot of people got UTI infections and isolated Salmonella infandis from that. Another outbreak from the pet food diet is a frozen mice outbreak. Okay, so we see a lot of salmonella infections associated with the feeder rodents. This happened in 2014. A lot of recalls happened on those feeder rodents. So these feeder rodents are something like freeze dry frozen mice. So they are in the market for primarily for feeding the pet snakes or reptiles. But people sometimes get it for to make their cats happy. By feeding frozen thing. So that's the reason why this may have a salmonella. It may not be an issue for reptiles, but it causes infection in cats. And a lot of people also got sick when they got the salmonella from this product. Okay, so we conducted a study in 2016 to look at some of those products in the market, and we were not able to find any salmonella, but we see a lot of other potential pathogens, and many are like antibiotic resistant. This is a recent outbreak happened last year and this year. We had saw a huge outbreak of a multi-drug resistant salmonella linked with the pig ear dog treats. Okay, so these are kind of dry treats available. Many of them are like imported, and you can give it to the pets, and pets really like it. Okay, so we see a huge salmonella outbreak, and most of them were like multi-drug resistant salmonella. They were found to be resistant to both the fluoroquinolones and the macrolides. These are the two first-line drugs we use in humans. So if a salmonella is resistant to that, most of the time, people are, will end up with the systemic illness and, with even like worse situations with hospitalizations because their primary drugs are not going to work. So we saw like 143 people infected, at least confirmed, and it spread across 35 states. And we saw like 26 hospitalizations, that was pretty high actually. And we banned that product now. The FDA put a notice, for people not to give any kind of, pig, any brand of pig ear treats. So FDA has got a website to list all the recalls and withdrawals. You see like, within the span of like 3-4 days, see how many products got recalled. Okay, and either with the Salmonella, Listeria, or E. coli, we see a lot of products now recalling, every day and this also put a lot of pressure, because a lot of products, these are like recalled products, so you can imagine how many samples might have tested to recall this many products. Okay, and FDA alone cannot do that now. Okay, so we have now an extensive system for testing and recalling, these products from the markets. So you can see that website updating all the products, recalls, in US. So the recalls are getting more and more common now, and but the good thing is it's now getting much faster. Okay, and now how is this made possible? FDA alone cannot do this. Earlier they used to do that, but now they are more relying on veterinary diagnostic labs, and we built a network system. All across US, we have now around 36 labs in US and Canada as part of this, network system. So we do test for this pet food diets, and we have, a whole genome sequence network. So any salmonella we isolate, in this diagnostic labs, we send it to the whole genome sequencing lab to get it sequenced right away. Okay, so that way, the system is now very efficient in tracking, diagnosing, tracking, and controlling any kind of pet foodborne infections. Okay, how the system work, so this is a lab network system, and FDA has a reporting portal, so this is an online portal where clients, or a veterinarian can go to initiate a client complaint, okay, that a particular product, is having, showed some infection or a diarrhea or any clinical sign, in the pets. And FDA will, follow up with them, and direct the client to send the samples to the network diagnostic lab, okay. And once that, sent to the lab, all the diagnosis or testing is going to be at free of cost. And also the labs will provide, information to the veterinarians to initiate a chain of custody if required, because that way those results are going to be valid in the court system. Okay, so but anyway, these are like all measures to make sure, like, if at all we find a issue with the commercial product, make sure the product get recalled from the market. So they have a detailed instruction how to report a pet food client. And so now moving on to some of those diagnostic, things we do for diagnosing this pet food infections. Okay, Salmonella, we do like a selective media culture for Salmonella. So it's one of the easiest organism to identify in the lab. And we do have enrichment techniques if the Salmonella are very low in numbers. We do enrichment cultures. Biochemical methods is a way to identify the bacteria, but we now moved more towards mold it off. It's a mass spec benchtop thing. You can identify the bacteria or the colony right away within few minutes. And once we have the bacteria, or with the sample, we can do a PCR or a selective enrichment PCR. That's a new methodology we developed. And once we have the isolate, we do like the antibiotic susceptibility testing to make sure it's not a multi-drug resistant Salmonella. And also we do whole genome sequencing on these Salmonella isolates through our network. So the diagnostic challenges we have, we do see a lot of different sample matrices. Sometimes, we get all these weird, rhodite things, and again, some of these are dry, food, or pellets, we have different matrices to process in the lab. That's a challenge for us, and then, the direct culture or PCR oftentimes is not sensitive enough because of the weird matrices, and even a lot of fecal inhibitors, PCR inhibitors, we may find in fecal samples. So even direct cultures, it's not that sensitive. So we developed a Salmonella enrichment culture, a combination of selective enrichment and PCR. Okay, the traditional Salmonella enrichment cultures are really good, but it may take 7 days to 14 days for direct enrichment or a prolonged enrichment to happen. But the selective PRV broth enrichment combined with the real-time PCR, we were successful to reduce the turnaround time to less than 24 hours. Okay, so if you get a sample today, we can report it out tomorrow. And at Cornell, we are doing it now routinely for a rapid environment or a hospital surveillance. Okay, people are concerned about Salmonella. Okay, so so the hospitals have now a huge responsibility to make sure, like the hospital environment is Salmonella free, like so that people can bring in their pets for even non-infectious causes like a vaccination. Okay, now think about the COVID situation. Okay, so people are worried, like to get infection, zoonotic infection, for their pets from hospital. So hospital now have to routinely test, for make sure like the environment is free of Salmonella. So we do this testing so that the turnaround times are much faster. So if they have a Salmonella positive dog, so they can ensure next day that their hospital is clean to accept more patients. So we also do serogrouping. That's a quick test we can do to kind of find the serogroup. So you have a rough idea about what the serotype might be. Then we can do serotyping, which is a little going to take few more days. And then we can do right away antibiotic susceptibility testing, and the whole genome sequence results will take another week time to get that. But all these are standard procedures associated with the any salmonella identification. So Molotov, this is a benchtop mass spec. This kind of revolutionized the traditional bacteriology nowadays. Okay, you can find it in almost every veterinary diagnostic labs now. And the good thing is you can identify the bacteria from the colony in less than 30 seconds. Okay, all you need to do is like with the toothpick, you pick a colony from the from the agar plate, put onto the steel target and one of those 96 wells, and the machine can identify it within less than 30 seconds. And it's highly accurate, is as accurate as a 16S DNA sequencing. And now labs cannot even think of working without this machine. Okay, and we have a full whole genome sequence procedure. Once we get the isolate, we do the whole genome in-house and get the sequences ready. Once we have the Salmonella whole genome sequence, we use as a FDA genome tracker database. Okay, this is a public database system, and we have curated whole genome sequences for Salmonella. We isolate from US and all over the world. Actually, a lot of people upload their whole genome sequences to this database. Okay, so this database is like very robust, and it's increasing day by day. And when I checked it last week, we got around 283,000 whole genome annotated sequences there. Okay, so this database is so powerful that we can now detect any foodborne infections happening anywhere in US very quickly. Okay, people constantly identify the salmonella, put to the system with all the metadata. So if you have a salmonella isolate, just blast it against this website. Right away, you can tell where else the same bacteria caused infection. Okay, if it's a multi-state infection, it can pop up within like a couple of days. Okay, because multiple people will get the same hit, and they can easily track the infection. Okay, so this is a very powerful tool available now, and we are more and more relying on this tool. We have Salmonella as the highest, but we are also building on E. coli, Shigella, and Campylobacter, and there are a lot of other, bacteria, including Staph student remedies. So this is one example where we used all these techniques to, help this client. It's a research facility at Rockefeller. They got like a salmonella infection in their SPF mouse facility. Okay. So these are research mice they use. These are SPF mice they use for research. And it's very unfortunate they get a bacterial infection there like salmonella, so that a lot of their research data got affected. So we got samples from the mice as well as the sterile, supposed to be sterile feed they feed, and we isolated Salmonella Livingston from both the mice as well as the feed. And we look for the single nucleotide polymorphism. If it is less than 15, those are considered as the same outbreak strain. Okay, so in this case, we were able to help the client, to make sure the infection is coming from the diet, and not from any other source. Interestingly enough, we were able to track based on the genome tracker that the infection may be coming from a meat and bone meal source, which they were using by the manufacturer to make the feed. Okay, so this is a very easy tool to source track the infection. So this is another case we use as the SNP method for source tracking brucellosis, especially Brucella canis infection in dogs in US. In this case, we got a Florida isolate. We were able to track that. The dog got infected from their breeder in Colorado. Okay, so the stud dog was in Colorado, and they were also became positive later. Okay, so we were able to source track the infection using this SNP whole genome sequencing technique. It's very common practice nowadays. So this is the slide on a Campylobacter. We also saw like Salmonella, Campylobacter jejuni can cause infection in dogs, but most of the dogs are asymptomatic. Only in puppies we may see some diarrhea. But in this case, we saw like a multi-state outbreak, with more than 118 people affected, affecting 18 states and 26 hospitalizations, when we do an EP study in this report, we find like more than 87% of people reported that they had a contact with the puppy they bought from a puppy store. Okay, so this ended up like a puppy mill issue, like, a lot of these people get puppies and they distribute across the US. And the unfortunate thing is this was a really bad bug, which was resistant to both the first line drugs, okay, the, macrolides and the fluoroquinolones. So that means a lot of people like, took those first line drugs, were not cured, ended up with hospitals, with more complications. Okay, so that's why this Campi jejuni infection really, was a big issue all across US. So like Salmonella, Campi jejuni also can cause infection, in puppies, and more importantly, it can, infect humans, and can cause a big problem. Listeria, we see like occasionally listeria cases in dogs and cats. This is a Bengal cat colony from Colorado State. We ended up with abortion. We isolated listeria from the fetal tissues, and then we realized that the cat was having a history of raw meat diets. And we were also part of the FDA investigation in a pet food recall in 2018, and this is the first time FDA took a direct action based on a diagnostic lab report. And since then, we improved our systems, and a lot of diagnostic labs are like routinely doing these things and helping FDA to recall the products. So talking about food safety principles to our pets, the same food safety principle applies, as in humans, like cleaning. Cleanliness is important, separating the food, chilling, and and about the cooking. Okay, any raw meat diet has a potential zoonotic pathogen, so it's better to cook the food for the pets. And now AVMA and FDA have directions not to use any raw ingredients in pet foods. And basically, the message is no raw meat diets for pets. And and One Health integration. This is a very good approach for controlling the zoonotic infections in humans as well as in for the public or for the owners. And my take-home messages are food safety is very important in animals to prevent zoonotic infections, and do not feed your pets with the raw meat diets. Companion animals can get infected with zoonotic pathogens like Salmonella and Campylobacter. And veterinarians and veterinary diagnostic labs have a major role in helping the clients to diagnose this disease, so tracking the infection, controlling the infection, and more importantly, to educate the client, okay, to not to feed the raw meat diet. Okay, you won't believe, like I give this lecture to the veterinary students since last 6 to 7 years. And every time, at least one student from a DVM student will come and argue with me that what I said is not correct. Okay, because they used to feed raw meat diets. Their cats have no issues. And it's not correct to say that not to feed raw meat diets. Okay. So I used to share my experience. One of my technician actually argued with me until she convinced later when her cats got really infected with a bad salmonella. And so even veterinarians sometimes used to argue against not feeding raw diet. So it's your responsibility to educate the clients to make sure like keep away from the raw meat diets. And finally, a One Health approach will help our pets as well as the public. We can both keep safe. And I would like to thank IVA and the Meat Technology Unit and the Meat Scientist Association for organizing this wonderful webinar. And also would like to thank Dr. Sunil, Dr. Maya, and B.C. Velayathan for inviting me and giving me an opportunity to deliver this talk. And finally, I would like to thank my teachers, students, fellow vets, and friends who's listening to this talk. And I would like to, wish you all stay safe and open for any questions.
Thank you, Dr. Anil Thachil, for giving in detail the problems of giving a raw meat to the pet animals and the diseases transmitted through that, which are of zoonotic importance, because CDC and WHO have declared in the list of zoonosis, the top 4 are Salmonella, Listeria, Campylobacter, Clostridia, and you dealt in detail on Salmonella, the latest the diagnostic techniques, and within 24 hours you can diagnose if a pet is coming, and based on that one you can admit to the hospital as well. And in poultry also, as I said, it's a little difficult to have Salmonella free stock because it will be expensive and may not be economically feasible as far as India is concerned. But still, I don't think here we have very few people give raw meat. Usually, they give meat, but not be over there. But by even improper cooking, then brucellosis and Listeria, Campylobacter, and they will hammer it these pet animals, and that can to human beings. So it's of vital importance. So very good. You elaborated on techniques and on the wonderful being in your North Carolina Diagnostic Animal Disease Diagnostic Lab. For anything you have for the record, it is not a practice over, but for that one you need the samples. So well-built lab should have. I might have questions from the list.
Participants are
Could you help out with a couple of questions from the participants and wind up the session, please?
Participants can ask question directly, or you can type your question in the chat box. You can just unmute your microphone and ask. Now I invite Dr. Deepa Jolly, Assistant Professor, Department of Veterinary Public Health, to sum up this session.
Hi, good evening
Dr.
to all
Deepa,
dignitaries. Yeah, am I audible?
Yes, yes.
Hello.
Yes,
Okay,
yes.
okay. Very good evening to all the dignitaries, delegates for this 3-day webinar series. It was indeed a pleasure to have co-chaired this session and on this topic on myths about the diets and zoonosis in pets. So actually, I would like to say that Dr. Anil, you were my classmate in our undergraduate years of study. I hope you remember me, right?
Yeah, I do.
So the thing is, it really was a very interesting session, especially on pet foods, because we don't much care about what is here, and we usually have cooked items here, what we usually feed our pets with. So about the session, you had covered some areas on different foodborne pathogens, especially the Campylobacter and Listeria Salmonella, and you're detailed on the CLSA activities, especially the veterinary breakpoints, vet 08 and M documents. So that was really something we didn't know about. So that also was an eye-opener. Now, some of the points here discussed is how diagnosing, tracking, and controlling this infection in pets was done in this. So we may also have to follow some methods like that. And also about the rapid and recent techniques, Mannuthy techniques that we use. That was really interesting. So I really thank you for having been here with us for this session here. And I just have one doubt. You were mentioning about the selective enrichment PCR used in Salmonella. So could you please elaborate the gist of what that is about?
Yes. Okay. So this is basically for environmental samples. We expect like very few number of bacteria there, and then again, it may have a lot of other inhibitors sometimes. Okay. So the enrichment actually, it's a, RV broth enrichment. So basically you get the, swabs or like kind of like, swift samples from the surface, you put onto this, large volume or 50 ml of RV broth, and then you incubate for 24 hours. Okay. So that, in even 16 to 18 hours. So that will, dilute any kind of, inhibitors and, selectively enrich this bacteria, and then after that, you have to plate it for routine culture. Instead, we do a PCR at that step, and we yielded like at least 100 times the more sensitivity than, doing a direct PCR on that sample or do like a culture. Okay. So that's an enrichment PCR technique, and now we were able to, give the results next day because hospitals cannot close their ICUs more than 24 hours. Okay, so if they got a salmonella case, next day they want to make sure like the hospital is salmonella free for admitting patients. Okay, so we do that routinely at the Cornell Hospital, because we don't know when a salmonella patient is going to end up, and we had to make sure like all the other pets are safe. So this is a very useful tool, so it's something like a mix of PCR as well as an enrichment, and better than a direct PCR. And you can see that manuscript is available online, detailing out the method. So it's in JVDI. You can it's open to public access.
Okay, thank you, thanks a lot.